Okay, now we're ready to start working on pandas. And this is the first really big open source package that you are encountering. And it is also probably one of the most useful and one of the ones that transformed Python and laid the foundation of Python as a data science tool. It was developed by Wes McKenney in 2008, and it has really transformed the way Python is used. Before pandas, you could not do process very easily in a fast way two-dimensional data like spreadsheet data, and this is really the bread and butter of what we need to do with very large data sets of millions of rows and millions of columns. And pandas solved that problem, and we'll talk a little bit more about what goes on under the hood in the last lecture about this. But for now, let's just use it as a library to access a relatively large data set and process it. Um, to talk about the data set that we are going to use a little bit, this is we are using the DepMap CRISPR data. It's one of my favorite data sets in bioinformatics. It's been generated by the Broad Institute in Boston. And you can go at the website actually and have a look what happens. But while this is loading, let's go just explain you the basic idea of it. So what they did is they collected data from cancer cell lines where each gene has been knocked out using CRISPR. And then after the knockout, they follow whether the cells die or survive, or how well the cell proliferates over time. And they repeat that for 20,000 genes, so it's a screen. They do this in one go, and they've got data for over 1,000 cancer cell lines. So it's a huge data set. After quite a lot of processing, you end up with a gene effect score. The more negative it is, the more the gene is essential. So if you have a negative score of -1, -2, that means those cells do not proliferate after losing that gene. Whereas if the score is around 0, that means the gene is not essential. It doesn't make any difference whether the gene is present or not. If the score is positive, it means the gene is actually preventing growth. And if you get rid of the gene, the cells grow better. So the data look somewhat like this. You have I collected that already and assembled it, so we download it straight from a repository that I generated. We have the model ID, which we don't really need to worry about. Then the cell line name, the lineage of where the cell lines derived from, so breast cancer or myeloid cancer, and then you get more distinct information on what this is an AML cancer, for example, and this is a breast carcinoma. So these first columns give you some basic information of what where these cells come from and what they're called. And then you've got about 18,000 rows with all the different genes from the human genome. And for each of these genes, we get a score. So in this case, the gene A1BG in the cell line HEL has got a score of 0.005. So this means this gene is absolutely non-essential. It doesn't make any difference to that cell whether it has that gene or not. And this gene here in this cell line, it's a little bit more essential. So this, but it's still not a very strong hit. So I hope when we're done, you'll appreciate how informative this type of data is. It's the only real data that give us a functional idea of like how important these genes are in different cancer cell lines, and that's ideal for drug companies, for example, to find new targets and things like that. Right. So now let's go back to pandas. This was just a little bit about the biology behind what we're looking at today. So what we need to do is we need to actually load the data in and then inspect them. And that's what we're going to do in the first notebook. We cannot, pandas is too large to run in the browser in a convenient way. So, so there's no more in the browser coding. We're gonna go onto the notebooks and work on the notebooks. So, but I just talk you through the code now quickly, the different commands, and then we'll go to the notebook and run the code. So the first thing is you have to import the library. So we import pandas as PD. When you run Python on your local machines, you have to do one more thing first. You have to actually download the package from the repository. But in the Google Colab, we don't need to do that. It's already pre-installed, but we still need to import it into the notebook. So we need to this line of code. Then what we need to do is we need to generate a data frame. So this is df, stands for data frame. This is normally what we call the variable that encodes the pandas data frame, but you could call it data or whatever. But df is a standard nomenclature. And this is the command. So with the pandas class, we can execute the method read CSV, and that will bring down the data from either from our local computer or in our case from a website and store it as this object data frame. And this is now what I showed you before, an object with superpowers. So this is like our hotel reception where we can go and with very simple commands do very, very complicated things that are all packaged in this df object. And then we can print it out and, for example, get the shape. And there's what we'll explore here in this first session is just some very simple and useful commands to analyze, to access this data. So you can see the shape, what are the column names, what are the data types. We can just get all of this information in one go using the info methods. We can describe statistics. We can check whether there's null values in there, and we can get the first few rows and things like that. So let's have a look at this now in real code. So we have to open the Colab notebook. And the first thing is, we import pandas. And then we're going to download the data from our website. So this is a web link to a repository that I made where I sort of cleaned up the data frames, the spreadsheets, so that they're ready to use. And they're also filtered, so we only have Brest and Mueller data on there. Otherwise, it would be too big. And this is already going to take a little bit of time to import because it's imported over the web, so it takes a little bit of time. You see, it's running, but it should come down soon. And then when it's done, we can explore with these first basic commands what this data look like. Fortunately, the internet is not going very fast, so you have to bear with me here. And is it coming? Right, while it's downloading, let's have a look at the command. So we get df shape, and this has run before, so I get the output already. So it's 94 rows so we only have 94 columns in this particular data set because we only have breast and blood cancer, and 17,211 columns, most of which are gene names. And this is when we write df head with the parentheses. So this is a method. It needs the parentheses. Then we access the first 5 rows of the data frame. You can see that it gives you the information, the cell line name, the oncotree lineage, the oncotree primary disease, then some subtypes. So this can be used for further filtering later on. And then come the main data, which is all the different genes with the dependency values. And we can also get the bottom of the data frame looking at the last row. So here's 93 to 89. Then to get more information on the structure and the type of data, we can just simply use the info method. So this gives us what are we looking at here? This is the object is a data frame object with 94 indexes, so 94 rows and 17,211 columns, and it gives us the data type. So most of the columns are float 64, like these data here. When it says object, it means the data are just strings or categories. And then the describe method is very powerful. It gives us an immediate summary of all this data. And as you sort of count the mean, the standard deviation, the quartiles, the maximum values. So this is very useful for a quick inspection and a quick summary. And then we can get, for example, all the column names, accessing the column attribute in the data frame. That will print us all the columns. And then to select individual columns, we use, again, like with lists, the square brackets. Then we just put the column name as a string into the square bracket, and that will give us the 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 one individual column. For example, here the model ID column, or if you want the this gene, for example, we just would go and put this gene here. At, I'd have to run this first. It should be run now, I suppose. Let's have a look whether this has run, and we'll execute. Yeah, so finally it's actually the download has occurred, so we can actually rerun all of these things. So this, once the download is done, everything else goes pretty fast. Yeah, so this is a first look at pandas. It seems like, oh, why can't you just use Excel? This data is probably small enough that you could just open it in an Excel file, but the whole data set will not load into where you have all the cell lines, where you have over 1,000 cell lines. This will not load anymore in Excel. And for this, we need to use a programmatic access of the data. So it's a very, this transition from working with Excel and seeing all the data in front of you to accessing data programmatically. It's a shift in mentality, it takes some time, but it is very powerful. So once you get used to it, you will not go back to Excel. I can promise you that. But it does take some time and some practice to get used to dealing with pandas and learning the most basic commands. It can get you very far very quickly, and we'll show you that in the next lecture.